important that we understand the thermodynamics by which metals or other substrates or cofactors bind to our enzymes.
That's good.
So from these techniques, we can get energy of binding, we can get enthalpy, entropy, and things like that. Dr. Wilcox is really one of the world's experts at using ITC and DSC to interrogate the binding of metals and other types of small molecules. And so, without further ado, he's going to talk to us today about calorimetry for bioinorganic chemistry, isothermal titration calorimetry, differential scanning calorimetry. Dean Wilcox from Dartmouth University.
Good. Thank you, Squire. Start by thanking Squire for the introduction and Karsten and all the bioinorganic team here at Penn State for inviting me to come. I'm delighted to be here because I view this as an incubator. I mean, you all are the future generation of bioinorganic chemistry, and the goal here is to give you information, tools that will be valuable for you throughout your career. And I'm thrilled to be here to share with you some of the knowledge that we've gained over the last 10, 12 years with these particular methods. Now, in bioinorganic chemistry, there's a lot of three-letter acronyms. We do EPR and NMR. We do MCD. We do XAS. We do DFT. And I'm delighted to say that ITC now has made it up into the big boy league. You know, it's up there with these big, heavy, powerful spectroscopic techniques. And DSC also fits with our three-letter acronym. So this is kind of a coming out party, if you will, for these techniques in our field. A point that Frank Nissa made early in the presentation was that it's important not to rely upon one technique only. So I do want to give everyone the caution that while these are powerful methods and they do provide some unique information, anytime you connect the data you get from these methods with other techniques, it gives you the reassurance that you haven't gone off on the wrong track. So that's an important consideration. My first day, I was sitting in the back row with Nikolai Leonard. Okay, Nikolai gave a beautiful talk yesterday with MCD. And he looked at the program and he said, "Oh, I see you're here. ITC, calorimetry, measure the heat, end of story. How are you going to fill 90 minutes?" So I just smiled. Because I'll do my best, but I think there's enough nuances in this method, enough potential problems, things that you really have to worry about. This is really kind of techniques for OCD, obsessive-compulsive individuals, who are really, really careful so that you can get data that's reliable and useful. Now, I've never given a talk for 90 minutes. I mean, I've got water here. I'll do the best I can. But I think the idea that the organizers had was that this 90 minutes would include discussion and questions, and we'd break it up. So I certainly encourage any of you to raise your hand, ask a question. If you have something that experience you've had or something that's not clear, please stop and we'll work with it. And so without further ado, let's go ahead and get started here. Okay, so here's just kind of an outline of what we're going to do. We're going to introduce some of these calorimetric methods, and particularly kind of the type of information they can provide for you. The bulk of the talk is on isothermal titration calorimetry. This is the one where we're measuring direct metal interactions with biomolecules. And so this is the one where it's cropping up more and more in the literature in our studies. We'll talk about the method. Specifically, we want to talk about issues dealing with metals. Okay, these techniques were developed, they've been around a long time, we'll see in a second, but they've kind of only recently been more used by people that are studying the roles of metals in biology. We'll kind of use a little teaching example here of this. Then we're going to look at several examples, some from my lab, some from others, where we'll kind of explore these topics, these ideas, and see how we can extract useful information with this particular method. And then I don't want to leave out at the end differential scanning calorimetry. Here's a technique where the metal is more of the metal contribution is more of a perturbation of the properties of the biological macromolecule, but you can get useful insight from that. Okay. All right, so calorimetry, what are we measuring? As Nikolai said, measure the heat. Okay, we're measuring the heat flow into or out of a system for some process, whether you're mixing two solutions, or as you see, we'll be heating solutions, and we're going to be measuring the excess heat that's associated with that. We're actually seeking the heat associated with a molecular or physical process that's taking place in that system, okay? But this is a bulk thermodynamic measurement. There are no quantum mechanics, no spectroscopy. Some of you may breathe a sigh of relief, no Hamiltonians, no projection operators, all of that. But the problem is here that you're measuring all the contributions to the net heat. So you got to be a real good scientist and carefully think about what's going on in the systems that you're studying. Okay? Now, if we do these experiments under isobaric conditions, then the heat flow is equal to the enthalpy of the process. These are the numbers that we're going to be very interested in. We measure the enthalpy, and we can determine the change in free energy for the process, which we can get from our K or KD, depending on if you're a chemist or a biochemist. Then, from our standard thermodynamic relationship, we can pull out the entropy. I have no idea what entropy is, but we can measure it. It's still a mysterious concept for a lot of us, okay? But this is probably, that's the core of it right there. That's what we're going to try to get to. Now, measuring the enthalpy is really the entree into thermodynamics. Okay? If all you're measuring is KD, you're measuring the equilibrium. Sure, yes, you're measuring the free energy, but I would venture to say until you get beyond that, you're really not into understanding the fundamental processes that are taking place. Okay? So we can measure the enthalpy directly, as we see with calorimetry. You can also measure it indirectly with the Van 't Hoff relationship, temperature dependence of the equilibrium constant. The assumption here, though, is that for the process, there's no change in the heat capacity. And for all biological systems, there is. So this is not a very good approximation for the types of things that we'll be doing. However, if you can measure this change in heat capacity, which you can by measuring the temperature dependence of the change in enthalpy, then you get an additional level of insight. This is, we don't really go even that far much in our studies, but this gets more at some of the entropic contributions of the processes. Okay, calorimetry. It's a pretty old method, okay? So we saw pictures of people from the early 20th century, Schrodinger, Heisenberg, Dirac. This is Lavoisier, a picture of Lavoisier's calorimeter. So think 150 years prior to that, late 18th century. It's in a museum in Paris. If I tried to pronounce the name of the museum, I would butcher it, so I'll just say it's in a museum in Paris. They've got Lavoisier's goggles, his apron, everything from his lab, including his calorimeter. Now, what is this device? It's concentric copper kettles, okay, with a little spigot at the bottom. And so what Lavoisier would do is he'd pack ice between the two kettles. He would do a reaction in the inner kettle, and as heat was given off, as most reactions are exothermic, it would melt the ice. He would collect the water from the tap at the bottom, and then he would weigh the water. So he made the connection between the mass of water that he collected and the actual heat that occurred in the reaction in the middle. Pretty ingenious for, 1780 or something like that. So calorimetry's been around a long time. Let's talk a bit more about some of the more modern calorimeters, okay? So many of them are designed to measure the heats of chemical reactions. And, we've got tables of these things, right? In fact, in our general chemistry laboratory, first-year chemistry students, we give them a styrofoam cup and a thermometer, and we say, measure the enthalpy of this reaction. Okay? Well, that's burning magnesium. You don't ask them to burn the magnesium in the styrofoam cup, because that would be a real mess. But by doing measurements in the styrofoam cup, measuring the change in temperature with a series of reactions, by using thermodynamic cycles, Hess's law, you can determine the enthalpy of this reaction from this very simple apparatus. It costs pennies. Okay? In more advanced physical chemistry courses, you may have done an experiment involved in bomb calorimeter. Okay? Device, very rigid material, So in fact, here now we're doing the reaction under isochoric or constant volume conditions. Okay, under those conditions, you're measuring the delta E. Usually this is for like determining the energy content of your breakfast cereal. Okay, you put in sugar pops and you blow it up in the bomb and you measure the heat associated with that. Okay. The two techniques we'll talk about are probably, I would say, fall more into the area of biological calorimetry. Okay, so these are techniques where we're going to be measuring the heats associated with biological molecules. We can't put them in a bomb, and we've kind of come up with ways to get at this information that are really quite ingenious. So, as the name says, isothermal, constant temperature, titration, so we're going to be adding things in aliquots, and we're going to be measuring the heat. So this is great for equilibrium processes, for binding, ligand receptor. These instruments were developed, the modern ones that have the sensitivity that allow the measurements we're interested in were developed in 1980s, 1990s, and it took really advances in thermocouples and engineering type advances to develop these instruments. But a few ingenious people said, let's develop instruments to measure these processes. Okay? We're doing under constant pressure conditions, so we're measuring enthalpy. Differential scanning calorimetry, differential, so we're measuring the difference between two things. Or scanning, we're actually going to be heating the sample up. Okay? Again, measuring the excess heat. This is particularly useful for looking at phase changes. Okay, now what do we think about a phase change? Well, a protein going from a folded to an unfolded state is a phase change. Okay? Membranes go into different phases depending on what's dissolved in the membrane. DNA unfolding. So this is a technique that is perhaps less useful for our field, but I think has a lot of potential. Okay, so these devices, okay? So we've taken devices that are about the size of a bread box now, okay? And this is actually a picture of the one that we'll be using in the tutorial, not this one in particular, that in fact allows us to measure this isothermal titration calorimetric measurements, okay? Notice it's computer controlled, so that's different than Lavoisier's time. Notice it's also encased in a glove box, so doing things under anaerobic conditions is very useful for certain types of measurements. All right, so that's the instrument. What are we doing? Okay, so inside that little box, okay, we have a reference cell and a reaction cell. These are about a mil. There are models that have about 200 microliter reaction cell. Okay, you fill your macromolecule usually in the lower cell, and then whatever you're adding in the syringe. Now, what's not shown here is all the amazing electronics that go into this. Okay? Very sensitive heating units, very sensitive thermocouples for measuring temperature difference, feedback loops and all. And so basically what we're going to do is we're going to set this thing up so that the outside of temperature is a little bit cooler than the temperature we want to work at. And then we're going to actually heat both of these cells just enough so that it maintains this constant temperature we're interested in. Okay? Now, notice we got this thing, a little arrow here, so we're stirring. We've got the little stir paddle in there. Okay, now, why are we stirring the solutions? Why are we stirring the solutions? Why do we have stir sticks out by the coffee machine? Yes, make it homogeneous, exactly. We want things to react quickly. Okay, we want to get that coffee, homogeneous so we can drink it, right? We don't want to have all the sugar sitting on the bottom, okay? So we're going to be stirring this because we want the reaction to go very quickly to equilibrium. We're less interested in the kinetics, although there are some interesting kinetics you can pull out of this technique, but we want to get quickly to equilibrium. So we're going to be stirring. So as we set up the instrument, we're going to get it stirring here because that stirring generates a little bit of heat, right? Okay? And so you want to factor that in as you equilibrate it. So these two cells are at the same temperature. One's being stirred, the other's not. Okay? And so we're ready then to start to inject a titrant. Okay? So we inject a small aliquot, a few 5, 10, 15 microliters. Okay? And what we're measuring over here is actually measuring the differential power between the two cells, but we kind of plot that in terms of microcalories, if you will. Okay? So we're going on equilibrium. We add our injectant. Okay? If it's an exothermic event, it gives off heat, so we don't have to apply quite as much power to our reaction cell, so we see a dip in the signal. Right? If the reaction is endothermic, the process is endothermic, then requires more heat, so we get a peak that goes in this particular direction. So this is kind of how we're measuring the heat that's associated with adding the titrant to our titrand solution, if you will. Okay, now, what about the titration part of it? Okay. So here's what the data looks like in kind of cartoon form. That's a good way to kind of start out looking at it as a cartoon. Okay, so we add an injection. Okay, it reacts, exothermic in this case, goes back to our baseline, okay, and we've added a small amount, okay, and now we're ready to add another aliquot, and we keep doing this, so it's a simple titration. Now, the first few aliquots we add, there's a whole bunch of the macromolecule in the cell, so they're going to react completely, okay, but as we go through the titration, there's less and less binding sites, and eventually we're going to run out to the point where we have this small background heat. Now, what's that background heat? Dilution, okay? We're still injecting titrant in, okay, but there's nothing for it to do except just to get mixed up, right? And so we've got this heat of dilution. Now, it turns out this heat of dilution, in fact, was contributing to each one of these peaks here, right? So you actually have to subtract that out of all of your data, so you don't include that in this bulk measurement of the heat that you do. Okay, so now look what we've got. Okay, so we've got these peaks. Okay, each one corresponds to a certain amount of our titrant. We're going to integrate the area of these things. Okay, we're going to include the concentration of our titrant. Okay, and so we're going to end up with a plot here, which is basically kilocalories per mole of injected. And now we're going to change our plot here instead of time. These are automated. Students love these instruments. Set up an experiment, come back in a few hours, and there's the data, you hope. Okay? But we're going to change our scale here now to a ratio of the concentration, the amount of titrant for the thing that's in the cell. Okay? So if we look here, our inflection point, where we go from binding to non-binding, in fact, then tells us our stoichiometry. Now, stoichiometry is kind of a soft number in calorimetry. Okay, because remember, you've got two different solutions. There's errors in the concentrations. Maybe not all the macromolecule is fully active. Okay, but stoichiometry is something that you can pull out of this. Certainly, you measure the enthalpy of this difference in heat from this fully injected to not injected. Okay, and then the slope here tells us about the equilibrium constant. Okay. Now, turns out, like any technique where you're measuring an equilibrium, you have a range over which you can do that. And in this technique, in calorimetry, it's known as the C window. I don't know who thought up this, okay? But basically, C is a number which is basically just the equilibrium constant times the concentration of the macromolecule. And so let's look at this C number. N is the number of binding sites, but let's just assume for one. So what we have here are kind of some simulations for C values where we're going to vary the K. Okay, we're going to hold N1. Here's the enthalpy, and here's the concentration of the macromolecule. And you can see that the data on the previous slide was probably, kind of somewhere in here, somewhere a C value of maybe between 10 and 100. And that was a pretty useful one. You could see some of these early points where there was a flatness here, and you can see that inflection. Now, as K gets larger and larger, notice that all the titrant gets bound until you run out of sites, and then all of a sudden you get a step function. Now, you approach that at about, 10 to the 8th, what's that, nanomolar binding affinities, okay? But that could be 10 to the 80th, and it'll be the same data. So really that sets the upper limit on what you can measure for an equilibrium constant. Going in the other direction, let's say your equilibrium constant is smaller, okay? Now you notice that some of the first aliquots, you're not binding all of your, now binding is occurring, okay? And you really have to go to larger excess to drive it. The curvature becomes harder and harder to fit to give you a useful number, okay? Now, we'll deal with a few cases where we have K values kind of on the edge of this, but trying to get data within this C window by adjusting the concentration of your macromolecule a bit, okay, is something that will allow you to get a good binding constant out of the data. Okay, so isothermal titration calorimetry of binding processes. We can determine binding constants over a range, okay? With the C window, probably a C value of like 1 to 1,000 is pretty good, but 5 to 500 is even better. So that's a good range. And so that limits this maybe a little bit less from this 5 orders of magnitude, but that's about the range that you can determine the binding constant. You also, of course, pull out the enthalpy, and you can back out these other types of numbers, so of parameters. So a lot of useful information, in theory, can come from one experiment. But we'll see that usually it takes more than one experiment to get the right conditions. Now, a few other things that might be of some interest to some of you at some point in your careers. One we'll talk about quite extensively today is proton competition. You know, protons are pretty important in a lot of bioinorganic processes, and this technique allows you to quantify protons released, picked up in solution. Okay? You can use this technique to determine reaction enzyme kinetics. This is techniques that were developed by this fellow Todd, who has done this early on. Recently, Gary Schenk from Queensland just published a paper where they use these methods to measure the enzyme kinetics of metalloenzymes. I won't go into that, but there's some references. You can also do this to measure redox reactions, okay, with redox couples. Okay? And Vern Parker, who actually was one of the instrumental in setting up one of the instrument companies, has this paper with Lance Seefeld where they showed one method of doing this. We actually are pioneering another way to pull out thermodynamics of redox reactions, and one of my students has a poster at the session, so if you want to learn more about that, stop by Molly Courteau's poster. Okay, so those are all good things, right? So, what to do with the data? So this is kind of the nuts and bolts of it, right? So the data you collect never looks like this. You know, you always publish the most beautiful data, right? The best data set, right? So it never looks like that. Usually, you know, the baseline is kind of looping, drifting. You know, it's hard to keep things, you know, constantly isothermal. You know, the peaks, you know, may or may not look quite as beautiful as that. So let's talk about kind of what you need to do. Now, this will be kind of the essence of our tutorial session, is kind of going over this, but just to kind of give you a little bit of a preview, okay? First thing you'll need to do is really kind of adjust the baseline, make sure that you're actually integrating all the area of the peaks that you've got there, okay? You'll have to subtract this heat of dilution, okay? Now, there are two ways to do this. I mentioned one where you look at this extended region out here, and that's your heat of dilution for the actual experiment. Another way to do that is actually to run a control titration, where you take the macromolecule out of the cell and just add your titrant, okay? It's not quite the same thing, okay? But it's useful to do that because you don't want to get fooled. Because sometimes when you add your titrant into a solution, there's chemistry going on. And you'd miss that if you didn't do that control titration. So that helps you avoid potential contributions. Okay? So subtracting off the heat of dilution, then you're going to integrate each of these peaks, and then you're going to end up with a data set that looks something like that. Okay? Now, it's good at this point to step back and do kind of a critical evaluation of the data. Okay? What about the injection peaks? Did I capture all the heat that's associated with all of that titrant that I added or not? Did I start the second injection too soon and I'm kind of missing part of that? So you may go back and change the time between injections so you capture all of that heat. Okay? There's kinetic information. Whenever I talk about this technique with hardcore physical chemists, they look at these peaks and they say, there's kinetic data there. You should be analyzing that. Well, you analyze it. I'm more interested in pulling the thermodynamics out of this. Okay. The stoichiometry. Okay. Oftentimes we have an idea about what the stoichiometry should be. You know, if your stoichiometry is you expect 1 and you get 0.3, you've got to go back and worry about things. It could be that, only 30% of your sample is active. You can still get useful data on that, but you want to try to make sure that this is all connecting. You want to make sure the data is within the C window if you're interested in pulling out the equilibrium constant. Some people use this technique only to measure the enthalpy. They get their equilibrium constant with some other method. Okay? So, if that's all you want, then you don't care if it's inside or outside the C window. All you need to do is make sure you measure all the heat. Okay? Now, what about that heat? Sometimes you can set up the experiment so there's so much heat you exceed the dynamic range of the instrument. Okay? And the peaks are cut off. Well, you got to go back and dilute it, right? But more often not, the problem is that you don't have enough heat, okay? That there's so little heat associated that there's pretty noisy spectrum, okay? Now, what do you do there? Well, the heat is an extensive property, so you can increase the concentration to amplify the amount of heat. Turns out by adjusting the conditions, you can also change the amount of net heat that you're measuring. For example, if there's protons that are in fact binding to the buffer, and you change the buffer to a buffer that has a higher heat of protonation, that can amplify your signal. We'll see an example of that. All right. Now, this is really kind of the trajectory. You get the initial data, probably it's crap. Even worse than that. Nothing happens. So you adjust the conditions. Eventually you get some data. It gets better. Finally, you get kind of optimize the conditions to get a good data set. And then you want reproducibility. Okay? These are very sensitive methods, and the errors between measurement are significant enough that you really need to have good reproducible experiments, 3 or 4 good titrations that are fairly consistent, and report the average values for that and the error associated with it. Okay. When my students give me data, I usually lop off several significant figures. Okay? Right. All right. Now, at this point, if you've got a good data set, now we're ready for a little bit of quantitative analysis. Okay? Oftentimes from the data, you can read right out the stoichiometry. You kind of get a sense of from the slope, what the binding constant is. You can actually estimate, the enthalpy associated with that. Not so with some data sets, perhaps, for example, weaker binding situations. So here comes fitting to a binding model. Okay? The most common one is just a single binding model, equilibrium. You may have done this in a course on thermodynamics. But there's software that fits these data, that provides these parameters. Notice N, K, and delta H, and the data software immediately just spits out a delta S also. Okay? I put that in parentheses because I caution us in the field working with metals that these numbers that come out from the fit, we have to do a little bit of what I call post hoc analysis. Okay? So I'm cautious about that number that comes out from just the fitting of the model. Now, if you have more than one binding event, then there are a couple of different ways you can do this. There's an independent binding sites model, okay, where the 2 sites fill in a statistical a ratio based upon their relative binding constants. Okay, and these models are generally available for 2 and even 3 independent binding sites. Another model is a sequential binding site, which sequential can mean n up to some high number. Okay. Of course, then you're going to get parameters for each one of these binding sites. Sometimes you may set up an experiment where you have a competition. Okay, so you may have a metal ion bound to one thing in your cell, and you're going to add in something that competes with the thing that's bound to. Okay, so there's a model that's generally available called this competition binding model. You know these parameters for one of the competitors, and you can extract it for the other. It's a little bit trickier thermodynamically because if the concentrations are changing dramatically, okay, then the activity's changing, and so this model takes into account. Okay, now this post hoc analysis. Okay, you do all this and you get a good data set and get these parameters. I still label them with ITC because these are still the experimental parameters that you have, and these are condition-dependent values for the pH, for the buffer, for everything. And you'd like to go further and get kind of more condition-independent values that really reflect what's going on at the molecular basis. Okay. Oh, I think, was it Frank? Somebody said, you got to reveal the dirty secrets, okay? So here it is. Early on, I don't want to scare you away from this method, but let's talk a bit about dirty secrets of calorimetry, particularly ITC. Okay, many laboratories have these instruments available from Microcal, now owned by GE Healthcare, or TA Instruments. The price tag is, expensive but affordable. So there's a lot of these around. Okay, some have been frustrated in their attempts to obtain useful ITC data. You are not alone. Okay. Some have published ITC data with errors in analysis and interpretation. They are not alone. Okay? So it's a bulk thermodynamic measurement. There's a lot of things going in, and you've got to be a good scientist to be able to extract out useful information. Okay, now, this frustration, okay? If you're frustrated, call me. I'm going to give you my cell phone number, okay? I'll talk you through it. I want people to get as much out of this technique as they can, but we've been frustrated. First experiment we did, I think, it was an experiment involving iron, and we precipitated rust inside the cell. Oh, what a mess. And the guy who owned the instrument almost shot me. It was like a disaster. So, but over the year, and actually it took us quite a bit of time to actually reproduce literature values. And, I thought, my gosh, what's going on? It wasn't until we put on our inorganic chemist hat and really thought about what was going on that we were able to account for all the things that are happening when you mix these two solutions and get good reproducible values. Okay, so some of you may have faced these situations. Okay, and hopefully we'll help you collect good data. Okay, so here, what are some of the issues here? Okay, these instruments are incredibly sensitive. I mean, we're measuring less than 1/10 of a calorie in 1 ml of aqueous solution. That's actually a phenomenally small amount of heat. So, they have good days and bad days, okay? So you have to work with it. You have to, in fact, yeah. All right. Contaminating species, okay? Care and cleanliness. Here's the OCD thing. You've got to be very careful. Of course, that's true with most of the techniques in this field, right? You got to be very careful. You got to be very clean. You got to avoid contamination, okay? Now, the big problem here is competing reactions and processes. And we'll talk about these with regard to metals. Basically, you need to eliminate them or account for them. It's really your choice. Okay? You can set up the experiment so that you can eliminate precipitation of the iron, right? So there's ways to get around that. Some of these things, competing things, you can't eliminate, so you just have to account for them in your post hoc analysis, right? And there are more complications when metals are involved. I talked to my friends in biochemistry who were looking at protein-protein binding or protein-DNA binding. The data spits out of the instrument and they publish it, okay? There are less complications. Us working with metals, it's hard, so. Okay, so let's talk about some of these issues with metals, all right? So this is really kind of the essence of what we're trying to do with a lot of the experiments we do with isothermal titration calorimetry. Okay? We're looking at this equilibrium, we're measuring this equilibrium constant, we're pulling out the free energy from that, we're measuring the enthalpy with calorimetry, and then we're pulling out the entropy. And then if we go one step further, we may actually be interested in the heat capacity. So this is it. All right? Now, here are the complications with metals. Okay? We prepare a solution, aqueous solution of a metal ion, with a buffer because proteins, our biological molecules, we want them to be buffered. By the way, this is a very protein-centric type of thing, but the same rules apply if you're studying metals binding to RNA or DNA or any saccharides, anything like that. Okay? So, buffers are Lewis bases. You have a complex usually with a metal buffer that you're delivering into the cell. Metals do redox reactions. We know all about that. Metals do hydrolysis chemistry, all right? And oftentimes this hydrolysis chemistry is dependent upon concentration. So you go from a concentrated solution of your metal in the syringe, you dilute it in the cell, and you got some of this going on, okay? You would only detect that in your control titration. You might not see it if you didn't do a control titration. So that's a good plug for doing that so you avoid all this. Now, there are ways to get around that, okay? Precipitation, okay, is another issue, right? So turns out if you can avoid the redox reactions, do it under anaerobic conditions. Put it in a glove box. Squire, yours is in a glove box, right? You saw the little boxes that we have for ours. So although you have to run it, 1 atmosphere of pressure, these experiments are open to the atmosphere. You can control that atmosphere, okay? Turns out these things can be avoided by delivering the metal as a well-defined complex that avoids hydrolysis and precipitation. And oftentimes the buffer can serve that role, okay? Sometimes you may need to add a chelate to make sure you know what the metal complex is that you're delivering. All right, this last one here, metals, oftentimes when they bind to proteins, they will displace a proton, competition between two Lewis acids, right? That proton goes then and binds to the buffer. There's heat associated with that. Sometimes that's the dominant heat that you're measuring. Okay? So these are complications. The ones in the middle are here, you try to avoid them, but this interaction with the buffer, you have to account for it. Sometimes it can be a friend. This proton displacement, you have to worry about and account for it, but sometimes it provides you with really good, useful insight. Okay? So these are the kind of things you have to worry about, the things that took us a couple of years to really kind of sort out in doing these measurements. Okay, so just a couple of summarizing comments here. The pH, or protons. There's going to be this competition with protons. Okay, couple equilibria involving protons are common. Need to account the heat that's associated with the proton binding to the buffer. So you can see the amount of heat will vary with the buffer. So that's why accounting for that is very important. Okay? The buffer, interaction with the metals, it can suppress these side reactions. It can provide a competing ligand. We'll see an example of that later on. We actually, therefore, need to know the binding constant, the metal buffer stability constant. We need to know the enthalpy associated with that, the stoichiometry. We need to know that type of thing so that we can factor it out in our analysis. Of course, buffers, we put them in to modulate the pH, but this enthalpy of the buffer protonation can be used to quantify the number of protons, okay, or even to amplify the signal that you measure. What about putting other things in the solutions? Oftentimes there's salt present, reducing agent, what else? Detergent, DMSO, all sorts of things, okay? Well, the key thing with regard to the titration is that the solution that you're adding from the syringe matches the solution in the cell as closely as you possibly can. So if you have to have salt, you want to make sure it's the same concentration in both your titrant and your titrans solution. Okay? Detergent, DMSO, these actually can be kind of tricky. Okay? But if you can match them, you have a chance of getting some good data out of that. All right? Trying to think, what else? What about reducing agents? Okay? Well, we have that so our protein doesn't become oxidized. Okay? But you're doing the experiment exposed to air. So you're doing the experiment, it's spinning away, okay? And air is leaking, your solution's in contact with air, and so your reducing agent is reacting with oxygen, and that's contributing to the heat. You don't really want to account for that. So if you can do it anaerobically, it avoids this. Sometimes you can put that in small amounts just to make sure that the sample is completely reduced before you start. But that can be an issue. Okay, so how do you account for all these? Well, so we actually do thermodynamic cycles. Okay, so this goes back, Hess's law, at least with regard to the enthalpy. So what do we have here? Well, we're starting out with a solution that has the metal. Some of it may be involved in a metal buffer complex. Maybe all of it is here, some of it here. Our ligand or our protein may be in different protonation states. Okay, so you have to account for that. You're going to end up with a metal ligand complex, and protons that are displaced are bound to the buffer. Here's what you're interested in. Okay, here's what you're actually measuring. So here's the KITC for all these things. So what we need to do now is, in fact, account for the things that we know we can account for, and hope that you've thought of everything. So certainly the metal buffer interaction, you need to know that. Subtract off that enthalpy because you're breaking that apart. You're deprotonating the ligand or the macromolecule. the protons binding the buffer, that's an enthalpy contribution, and here's the thing you're interested in the end. So these thermodynamic cycles allow you to pull out that number. That's what you're interested in. And you can do the same thing for the free energy or analysis using the equilibrium constant to account for other equilibria. Okay. All right, so let's do a few examples. That's simple case. We're going to look at nickel binding to histidine, just to kind of run so quick examples of this. Okay, so forms 1-to-1 and 2-to-1 complexes. Okay, here's the two equilibria that we're interested in. Okay, so here's a titration: nickel into histidine into nickel. So we're adding the ligand into the metal. We expect to see two binding equilibria. Okay. Because initially we have a whole bunch of nickel and we're just adding little bits of histidine. So we're going to form the 1-to-1 complex initially, okay? And then we're going to form the 2-to-1 complex. We see how we have a major inflection here about 2, but notice the very first injection had somewhat less heat, and then the heat got larger, okay? So what does that tell us about the enthalpy of the first step versus the second step? Which is less, which is more? Less heat with the first aliquot that goes in, which is this first step, right? So it turns out this second binding event actually has a little bit more heat associated with it. So it's not a flat baseline, okay? It's not flat along here. If it was, that might suggest we had kind of cooperative binding, okay? And all we were forming was a 2-to-1 complex throughout. But this kind of small difference in the baseline gives us a clue that it's a sequential binding event. All right. Now, with this case, we can actually turn things around, and we can put the metal and titrate it into. And so now what we've got is a different situation. We have a large excess of the histidine, and we're adding the nickel. So the first thing we form is the 2-to-1 complex. Okay? And we notice that our big inflection here is at about 0.5. Okay? And then as we add more nickel, we now form the 1-to-1 complex. Now, not much heat associated with that second process. It looks like it's a little bit endothermic, in fact, right? What's going on here? Well, to go from here to here, you have to remove one histidine from one nickel and put it on the other. It's kind of like isoenthalpic, if you will. So there's not much total heat for that particular process. So we're learning something about kind of sequential versus cooperative type of binding in this. Okay, but to pull out the numbers, okay, in fact, just for the first binding equilibria, we've got all these things we've got to worry about. So let's just kind of look at these kind of independently. First of all, let's look at the buffer effect. Here's this same data. So histidine into nickel in Heap's buffer. Okay, and there's the data we saw before. Here's the same titration. pH is a little bit different, but it's in phosphate buffer. Now the net thing is endothermic. What's going on here? Okay, it's essentially the same binding phenomena. We're just in a different buffer, a little bit different pH. Turns out that what the main difference is the enthalpy of protonation of these two buffers is very different. Okay, and remember, when the nickel binds to the histidine, we're deprotonating the terminal amine, so there's a proton that's released. And so much of this exothermic signal here is the protonation of the Heaps. Protonation of phosphate is very, very small. Okay, so buffer heat matters, okay? So it can often dominate, and you have to account for that, okay? What about the nickel-buffer interaction, okay? Well, you can do this experiment in a variety of different buffers, okay? And as long as you account for the nickel interaction with the buffer, which was done pretty rigorously here with tris and phosphate, you come out with K1 values that are pretty darn close in the overall equilibrium constant. So you really do need to account for this. Now, sometimes this is pretty small, and it's a small contribution, but it is something we have to worry about. Okay, now what about protonation states of the complex? Well, this is kind of summarizing up the whole thermodynamic table, but basically we're looking at different protonation states of histidine and nickel-tris complexes. But here's kind of a little thermodynamic table you can do. Here are the values that you get from the overall experiment. Here's accounting for the individual steps, and you can see when you come out with it, you end up with values that are pretty close to the literature for the equilibrium constants. Okay, so you really do need to account for all the complexes and the protonation states. Now, don't be overwhelmed by this. It's fairly straightforward type of analysis, and we'll see that in many cases it's more simple than this. Okay, so let's do a couple of cases. Let's bring in some old friend proteins, and look at metal binding to them, and see how some of these tools that we can use to extract information about the metal binding. So these are the examples we're going to consider. A couple are from labs of some real pioneers in the field, and some are from our lab, and we'll talk about these kind of individually as kind of teaching examples, if you will. Okay? So here's transferrin. It's the iron transport protein in our blood. Consists of similar N and C terminal domains. Each one binds one ferric iron with a synergistic carbonate anion. And these pictures from Lipperdenberg's book show the structure of the apo form and the structure of the metal-bound form of the old Pac-Man type model. Chomps down on the iron. Right? Here's the iron coordination, very well studied. Binds iron 3 fairly tightly, so the K-parent value is way outside of our C window, right? Okay, so this fellow, John Brantz, is one of my heroes because he was one of the founding fathers of biomolecular calorimetry. He founded MicroCal, and his guidance made that a company that provides a very nice product. And these papers here are really the seminal papers in how to use calorimetry, ITC, to study metals binding to proteins. Okay? And it was work done with Woodworth and Mason up at the University of Vermont. And so they did calorimetry of iron binding to transferrin and ovotransferrin. Notice they're delivering the iron as the NTA complex. Okay? So what we're going to have here is a competition between the NTA and the protein. The protein's going to take the iron away from the NTA, but that's a way to solubilize the ferric iron and actually get an overall equilibrium constant that fits within our C window. Okay? We look at the injections. Okay? So here is a controlled titration, okay, of iron NTA just in solution. Notice it's endothermic, so nothing funny going on here. Okay? Here's our titration with human transferrin. We see nice injection peaks, okay? Then they start to change shape, okay? And then eventually we get to our controlled titrations. So basically what's going on here is the iron is binding to these two different domains of the protein and with slightly different thermodynamics, okay? As indicated by the different peak shapes associated with the first one binding site. This is with human transferrin. So it's binding to the C domain first and then the N terminal. Okay? Here's the data for ovotransferrin. Notice the peak shapes are significantly different here. But again, we get this kind of transition from one domain to the second one and then our control. Okay? And so analyzing this data, and Brandt's paper lays it all out. None of us have done anything different from what he did back then. It really is a good model. Okay? He found that the equilibrium constants measured the enthalpy. Turns out this enthalpy is very temperature dependent, which means that it has something to do with the heat capacity. And in fact, by careful analysis of these data, they were able to figure out the thermodynamics of interaction between these two irons at the two sites that are about 40 angstroms away. So some really a very good model paper. Okay. Ferritin. Okay, the iron storage protein, right? Seen this picture? This is from Jimmy Cowan's book. We've got this protein shell. We got the iron binding in the core as a ferrihydrate mineral. Okay, two irons bind initially to ferroxidase sites. Okay, where they're oxidized to form the mineral core. Okay, and the release of the iron involves labile iron 2. So here, what you're going to be interested in is the iron 2 binding phenomena. Okay. Anybody worked with iron 2 here? Yeah? You got to work in a glove box, right? It's oxidized pretty easily. So this was experiments that had to be done under anaerobic conditions. And so this is data from Dennis Chastain's lab at the University of New Hampshire. And so let's look at this data for just a second and think about what's a little bit different from the data we've seen before. How about signal to noise? Yeah, it's kind of noisy. Okay, Dennis makes no apologies about this data. We'll see why it's a bit noisy in this case. What about the units? Joules. He's a physical chemist. He's using the right SI units. The rest of us are using these old calorie units, okay? Also, notice here that these peaks look like they're exothermic, but then when they're plotted here after they're integrated, they're endothermic, okay? Turns out he was using another instrument that was earlier company called Calorimetric Sciences Company, and they said their instrument was set up a little bit differently. And so down peaks mean endothermic, up peaks mean exothermic. But basically, in the end, what you get is, in fact, that this binding is endothermic. Okay? Now, another thing to notice. What about this stoichiometry? Pretty large. 50 equivalents of iron 2. Okay? And in fact, when you fit this data to a one-site model, okay, all sites are equivalent, the N value is 23. So a lot of irons are binding, all with equivalent thermodynamics, as best you can tell from these data. And it is endothermic. Okay? So you can tackle some pretty tough problems. This had to be done anaerobically. It involved, some fairly rigorous work done by Dennis and his postdoc, Fadi Abu Abdallah. Yeah.
So what does this N value of 23 actually mean? I mean, is there a plus or minus that goes along with that? Can you trust that information?
Yeah, there should have been. There may have been in the paper. I didn't take it out.
But like from your own sort of dealings with these because
that's a
have N values that we don't expect, right?
Yep. And the question is, is it something I did wrong, or is it true, true N value? Yeah. Yeah. Here. Yeah. Now that raises an interesting point. When you fit these data, it's going to give you some parameters with an error associated for that particular experiment. You do the experiment again, and you get error values associated with that experiment. Okay. The difference in the values leads to an error that's larger than either one of those. So that's why you really have to do multiple experiments and then take the average. Okay? So I don't know how many times they repeated this, what the error is on that particular number, but that's a very good question. N is soft. It's a soft number. Okay? You really got to work hard to get a...
Unless you really know it, then you can put that in the fit.
Exactly.
Exactly.
And we'll see in a second that knowing it by another method can really save you. Okay? And so, good question. All right. Albumin. So this is the protein we're going to use in our tutorial because it's pretty robust. It binds all sorts of things. Okay? It's an interesting protein, like 17 disulfide bonds. Okay? One free thiol here. It's cysteine 34 up there. It binds and transports copper nickel at this N-terminal XX his binding site. And copper binding to that site is what we're going to measure in the tutorial. Okay? Now, here's the crystal structure, and there's lysine 4. So you can't actually see these amino acids in the crystal structure. They're disordered. They're kind of dangling off here. To look at the sequence from a whole variety of species. Most critters have histidine in this third position. A few don't. Okay? And here's the coordination that you find. I guess it's now called ACTUN, amino-terminal copper-nickel binding site. It's got its own acronym now. All right? So what we have is the metal is binding to the imidazole of that histidine in the third position. It's binding to the terminal amine. And it's also deprotonating these two amides. Now, copper and nickel are about, they're strong enough Lewis acids that they can do that under physiological conditions. Other metals don't. So that's why this site is specific for these two particular metals. Okay? So that's the coordination that we're interested in. Notice what do we have in terms of protons, right? Well, usually the amine is deprotonated, right? Is protonated, so it's going to deprotonate that. You're going to lose 2 protons. So when the metal binds here, as long as you're above the pK of histidine, you got 3 protons that are going to be displaced when the nickel or copper bind. Okay, a lot of measurements have been done on copper and nickel binding to this protein by a variety of methods: dialysis, ultrafiltration, ion-selective electrodes, different conditions, competing ligands. Here's the column of interest. Here's the apparent Ka. It's pH 7.4 log value. So that's a very high stability constant. That's way outside of our C window. How are we going to deal with this? Okay. Well, it turns out copper forms a pretty stable complex with tris buffer. So we can now use if we do it in tris buffer, we can use the buffer as our competing ligand, and it's a competition between copper binding to tris and copper binding to the protein. And there's the data. Now, this is at somewhat higher pH. For some reason, I know why, we were working at somewhat higher pH. But here's the data. Copper binding and we've got tris present. And so actually, that's a pretty nice binding curve, right? You could fit that. We did fit that. Stoichiometry is about right, 1-to-1. You can plot a binding constant out of that. Now, remember, this is competition between tris and the protein. You can pull out the enthalpy of that. So one can do an analysis of that. Now, I mentioned this one cysteine, okay, this one lone thiol. Turns out you can prepare albumin where that's reduced and free or oxidized, okay, or blocked, reacting with N-methylmalonide, okay? And so you can take measurements and, in fact, do the analysis, okay, simple analysis here. You have to factor in the competition with tris. You got to factor in 2 protons. Now, we're up at high pH, okay, so we only have to worry about the 2 amide protons. We're going to include them in the analysis. These numbers are a little bit funny because of that. You know, we don't have a working pKa for that. But we can actually get the log K at 7.4, and this falls right within the range of what was measured by other methods. So we can pull out a binding constant, but remember, we're getting the enthalpy out of this measurement, okay? We notice that the stability varies depending upon the oxidation state of this cysteine. Talk about that in a second, okay? But when it is reduced, notice what's different here. The enthalpies are pretty similar, but there's a pretty large entropic difference. So this difference in affinities here, almost 1 order of magnitude, really boils down to an entropic difference. So that's kind of useful insight. How does that relate? Okay, well, what's going on up here about 20 angstroms away is in fact affecting what's down here. So there's some entropic, predominantly protomic factor that's in fact influencing the binding thermodynamics of the copper. Since this was pretty easy to work with, we actually went ahead and measured the temperature dependence of the enthalpy for copper binding. And by the slope of this, we pulled out the change in heat capacity. Now we get some numbers. What do those numbers mean? Let's compare them to something we actually just saw. Okay? So I mentioned transferrin that Brantz studied, okay, and they looked at the temperature dependence of the enthalpy, and for that case, they got pretty large negative changes in heat capacity. Well, that kind of makes sense. You know, the protein's going from this open Pac-Man formation to a closed formation, so a pretty large change in the protein structure and therefore its interaction with solvent. Okay? And this large negative number is in fact generally associated with large changes in the protein structure, in this case associated with metal binding. But here, remember, these amino acids are kind of dangling out there. You can't even see them in the crystal structure. So the binding, the change in heat capacity, is not very big. In fact, it's positive, and it really relates to the metal coordination to these 3 amino acids. And it's small and positive. Okay? So we learned something from this, that big changes in the properties of our macromolecule, solvent effects, that's where we get big changes in our heat capacity. Okay, another story. My colleague at Dartmouth, Mary Lou Guerneau, actually discovered this protein, okay, called IRT1. It's a membrane protein in the roots of plants. It's involved in metal uptake, predominantly iron, since that's the limiting nutrient for plants. Here's the protein. It's got 8 transmembrane helices. She's mutated different amino acids and looked at how that affects metal transport through this protein. But what caught our attention was, in this large intracellular loop, there's this sequence, P, HG, 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 HGP. You know, when nature does that, it jumps out at you. You say, whoa, look at that regular repeat pattern. Look at that high concentrations of histidines, metal binding site. Well, it's a little bit hard to study membrane protein. Remember, this loop is inside the cell. So now I think we're developing techniques that you can actually measure this types, do measurements inside the cell. But this is a little bit tricky to study the metal binding properties of that sequence of that loop. So we synthesized the peptide and looked at its metal coordination properties. And it turns out it's an instructive little exercise to study it. Okay? So the first thing we did was zinc. Okay. Now, actually, a lot of the studies we do involve zinc. You can't do MCD on zinc. You can't do EPR on zinc. Okay? You can do X-ray absorption on zinc. Okay? But zinc is one of those D10 metals that we don't have as many tools as we'd like to. So it turns out calorimetry is oftentimes a way to provide some good insight on these spectroscopically silent metals. Okay? And zinc's an easy one. You don't do redox chemistry on it. So here, this is just zinc binding to the peptide. Okay? And here's the binding curve. So if we look at that and think about it, you know, that's not our usual kind of sigmoidal type shape. That's a fairly weak binding, right? And there's the binding constant we get out of it. Okay? Notice we went out to large molar ratio so that we could get a good isotherm to get a pretty good fit to that. So that really helps you, subtract off the correct dilution to give you a good value. Okay, and notice that in these weak binding cases that you can't actually read off the enthalpy. You don't have this initial flat portion where you're getting all the heat binding. So the enthalpy really has to kind of come out of the fit to the data. And you can see here's what the fit predicts, is the enthalpy is actually pretty small. Okay? But we can pull out the binding affinity for zinc with this particular case. Now, here's an interesting one because that was done in ACEs buffer. If we do this binding in different buffers, we get different enthalpies. And from that, we can actually determine how many protons are bound to this peptide at pH 7.2 that are displaced by the metal when it binds. Okay? So what we're going to do is here's the enthalpy, and here are the main contributions to it. Okay? What we're interested in is this little n here, number of protons multiplied by the buffer protonation enthalpy. So we're going to plot the experimental enthalpy minus the metal buffer interaction, because that's going to change as we change the buffer, versus the buffer protonation enthalpy down here for a variety of buffers, and we get a nice, pretty straight line. We pull out n of 0.79. So about 8.8 protons are bound to the peptide at pH 7.2 that are displaced by the metal. So we did that in solution. Okay. All right. Now, this protein is involved in binding, transporting iron. So we better do some experiments with iron. I told you my story about rust, precipitating rust in the cell of one of our first experiments. And it turns out that iron, we predicted, binds pretty tightly to this protein. Turns out iron 3 is a little bit tricky to work with. As Brantz showed, he had to introduce it as a NTA complex. But here's an alternate way to measure metal binding. We call it chelation measurement. So we're going to start with iron bound to the peptide. Okay. Notice we have a pretty large excess of peptide to make sure all the iron 3 is bound. And then we're going to titrate in EDTA, which has a higher affinity than the peptide. And so we're going to chelate the metal out. Now, to do the analysis here, and we get a pretty good binding isotherm, right? Okay. Right stoichiometry. This is based upon the iron. Okay. We get out the binding constant and the enthalpy. But remember, this enthalpy also includes the iron binding to the EDTA. So we've got to factor that out if we want to back out the value of the iron 3 bound to the peptide. So we're measuring kind of the dissociation enthalpy, if you will. And another beautiful example of this is on Molly Carpenter's poster out on the walkway here, where she did this type of experiments to measure zinc binding to insulin, where insulin is unstable in the absence of the zinc. So you got to preform it and do it in a way to measure zinc bind out of that. So this chelation experiment was a way that we were able to get data for iron 3, which is tricky otherwise. Iron 2, okay? So, in the plant it could be reducing environment, inside the cell, reducing environment. So what about iron 2 binding to this peptide? Now, let's see. Strictly anaerobic conditions. Look at the molar ratio here, okay? Look at the number of peaks here, okay? Turns out this was 3 syringefuls of iron 2. This is a process it's called concatenation. So you do one experiment where you collect data, I think, up to about here, okay? And then you take out the syringe and you put in more of your iron. The system will equilibrate, and then you continue on with the titration. And so we got, we're able to titrate out to almost, 25 equivalents of iron, and to get a binding curve. Now, why was that necessary? Okay, well, look at the binding constant here. That's pretty darn small. That's way outside the C window. Okay, how do we believe this number? Okay, well, notice here, N1 is fixed. Okay, we know that this peptide binds metals 1-to-1 stoichiometry, so we're going to fix that, and that then allows us to vary just the binding constant and just the enthalpy. We got this very extensive data set, and we get a pretty good fit. So you can actually pull out binding constants that are way low on this C window if you know the stoichiometry and you collect enough data. So there's hope in that case. Okay, so looking at a variety of metals, okay. On Wednesday, Frank told us about ligand field theory, told us about the double hump, right? The experimental proof. Well, turns out here we've got it. Okay, we've got ligand field stabilization energy shown here with this peptide across the first row. This is only half of the double hump, of course. Here's the free energy, okay, going negative, manganese to zinc. Here's the enthalpy, okay. Notice that double hump is predominantly enthalpic process. Here's the entropy. I don't know that this data is actually in the literature anywhere. What is the entropic contribution to this binding in a periodic sense across the first row? And notice that it's same hump, but it's the opposite sign. Okay. Here's iron 3, very high affinity. We pulled that out. So if you are careful, you can go ahead and get this type of thermodynamic information. Okay, metallothionein. See, when ever heard of metallothionein? Very unique protein. It helps save us if we're exposed to toxic metals. It's got about 20 cysteines, binds a whole variety of fairly soft metals. Zinc, it's believed that it may play a role in zinc homeostasis. I'm not sure I believe that, okay, but that's a hypothesis out there. So a lot of people have studied zinc binding to metallothionein, in addition to, of course, these toxic metals that bind all the cysteines in metallothionein. Now, it binds a lot of them, okay? In fact, here, this is, I think, the structure with cadmium. We've got 3 bound in this domain and 4 bound in this domain. And through some interesting spectroscopic methods, mostly fluorescence, Merritt was able to, in fact, measure some equilibrium constants for zinc binding to this protein. Well, we decided to tackle this with calorimetry. Now, this was a daunting task, but Colette Quinn, who now works with TA Instruments, was up to the challenge. So here's data for zinc binding to this thiol-rich protein in different buffers. Notice the data is qualitatively different, okay? But there are some similarities to it. They all seem to have kind of 2 events, 2 inflection points here, okay? And if we set tris aside for a moment, these 3 actually have kind of fairly similar, it's, 3 to 4 in the first event and 3 to 4 in the second event, okay? Well, Colette analyzed these data, okay, and under a model where we have 4 initially binding and then 3 binding subsequently, and fit the data in the different buffers to pull out the thermodynamics of this particular process. And it turns out by doing it in the different buffers, she was actually able to show that there actually, when the first 4 zincs bind, you displace 12 protons from the thiols. But then when you add the second set of 3 zincs, a lot fewer protons are displaced. Those initial zincs go in and bind and displace most of the protons. The second zincs come in and they form bridging positions. So the protons are already gone. So this actually quantifies the thermodynamics of that particular binding. It was, kind of a tour de force to pull this out. Okay. Binding of other metals. Okay. Here's the zinc binding in tris buffer. Here's arsenic binding in tris buffer. Qualitatively, you can see different binding events. This data is very indicative of identical binding sites for arsenic. It's always the same enthalpy all the way across until you, in fact, bind. So it's kind of like a big polythiolate ligand for arsenic. But for zinc, we do find two different types of binding thermodynamics associated with that. Now, it's tough to work with a protein that has 20 cysteines in 61 amino acids. It's really tough when you're working with the apoprotein. So another way to get this type of information is to do something called a metal displacement experiment. So since we know the zinc binding thermodynamics, and we know that metals like cadmium and lead will displace the zinc, you can titrate in these tighter binding metals, and you can, in fact, find binding isotherms for this displacement experiment. Notice that the buffer, in fact, has a pretty dramatic effect has a noticeable effect, if you will, on the cadmium binding, less so on the lead. Here's a caution with lead, you got to be careful. You got to know the metal chemistry, okay? So lead at neutral pH has a lot of hydrolysis issues. So you work at slightly lower pH and you can get reasonable binding isotherms. Okay, the last story I want to tell, okay, is an interesting one. It was a collaboration with Bob Hausinger up at Michigan State. He has studied for years urease. We did also. He's looked at the process by which nickel is inserted into urease. So the pathway by which you get the right metal in the right metalloenzyme, okay? And all the proteins that are associated with that are associated with URE operon. UREE is the protein, the metallochaperone, that delivers the nickel to the urease apoprotein. So it plays a pretty important role. And so Bob has studied this, and he actually prepared a slightly truncated version. This protein binds 2 nickel ions with different affinities. This was done by equilibrium dialysis. It also binds 2 coppers, which has been characterized by kind of some of our techniques we know about now. Okay, and then he did some crystallography with the Huber group. And it showed the structure of this UREE protein. And we got together and started to study the metal binding by calorimetry, because this is a metal transport. It's got to bind the metal, it's got to release it. So binding that's its function. It's pretty important. So here's the crystal structure. It actually has 2 domains. I think it said there, one is an ATX type metal binding domain, and the other's a heat shock protein. So it binds as a head-to-head dimer. So these are the 2 ATX domains that are bound here. So there's the crystal structure of it. And then what they did was they soaked copper into the crystal. And then they did crystallography again. And what they found was 1, 2, 3 coppers. Equilibrium dialysis said 2. We got a discrepancy here. So what's going on? Well, here's where calorimetry came to the rescue. Say modestly, all right? So here's copper binding to URE protein at 6 micromolar concentrations. This kind of gives you a sense in the calorimeter, protein concentrations, tens of micromolar, and then you're titrating in the metal ion, okay? And we look at this binding data here. See nice inflection about 2. We pull out the thermodynamics, 2 copper ions binding to this protein, okay? Increase the concentration to 25 micromolar. When Nick came in and showed me this data, I said, Nick, what's going on here? He said, ooh, this is going to be fun. And he looked at this data and said, look, you got 1, 2, you got 3 binding events going on. And you look carefully at it, and you see that up here, the peak injections have slightly different shapes. Some of them have this little endothermic peak here. So clearly, you got a much more complex type of binding phenomena when you have this higher concentration. Okay? Now, fitting it was a little bit of a challenge, okay, because we only had a 2-site binding model, and here's 3 events. But you could actually fit different parts of it, so you can fit the first 2, you can fit the second one, and basically you can fit this out. And so now, these are stoichiometries per dimer. The data here is per tetramer, but this is stoichiometries per dimer. So you get 1 binding per dimer, or half of a metal binding per dimer, or 1 per tetramer, another half per dimer, okay, and then you get 2 more per dimer. So this ends up with 3 per dimer, or 6 per tetramer, okay. So what's going on here? Okay. Well, here's the model that we came up with to explain that situation, okay. So at low concentration, all you have is a dimer, and so all you have are these 2 periphery sites, okay. So you get 2 binding per the dimer, okay. But as you go up in concentration, now you have a higher aggregate state. You can actually have a tetramer, okay, or you can bind 1 at this interface site, you can bind 2 at this interface site, and then you can bind them at the periphery. So what this turns out to be is what you saw in the crystallography. For a dimer, you've got the 2 that you probably are measuring by equilibrium dialysis, but then you've got this additional one that's binding at this interface of the tetramer. So that kind of sorted out a controversy in the literature. Okay, and we pulled out the thermodynamics of this. Okay, so kind of to summarize up these kind of examples, what we've kind of pulled out of using calorimetry. Okay, so in general, you need to know the metal chemistry, okay, if you're going to be doing this technique, because of all the competing reactions. A perfect example is studying copper-1. Okay, copper-1, like zinc-2, is D10. It's, again, not so generally accessible by spectroscopic methods, but it is, the oxidation state that's probably dominant inside the cell under reducing conditions. Okay? So you got to be careful because copper-1 disproportionates. And you got to prevent that because over the course of your titration, you'll be maybe measuring the heat of disproportionation. And that's not what you're interested in. With albumin, okay, we saw that if you deliver the metal as a well-defined complex, a complex that can suppress that type of chemistry, okay, with the right types of ligands, you can stabilize copper-1. That avoids unwanted reactions and allows you to measure high-affinity binding sites by this competition. Always want to compare it to other measurements of the equilibrium constant. This is a way to connect to other data, other types of measurements. With this peptide, we, in fact, saw an example where we quantified the number of protons that are displaced. We also saw that you could measure metal binding by this chelation, by measuring the thermodynamics of ripping it out of the protein. Okay. Low affinity, though, you need to know the stoichiometry. Multiple binding sites requires analysis with different binding models. Okay. And finally, you can deconvolute some complex isotherms, but it requires accurate stoichiometries. That was really kind of the key thing there. By the way, Nick and Ann Spuchis and I wrote this review article. It's kind of a good, useful thing to have. A lot of tips and things that are summarized here in that particular article. Okay, I'm going to skip over this. I didn't know if I was going to fill 90 minutes. Nikolai threw down the gauntlet, and so I threw this study in, and we really are probably, it's just too much to go through. We did some studies of zinc binding to zinc fingers and pulled out some very detailed thermodynamic information, some interesting insight about enthalpy-entropy compensation. This is something that, calorimetrists get excited about, okay? But I want to spend the last few minutes talking a bit about differential scanning calorimetry. Okay, so this is kind of the other technique that we at least are starting to use to study metals. It turns out, I mentioned this seminal paper by Brandts where they studied iron binding to transferrin. They also did DSC measurements on that too. So there's a companion Brandts paper that you can look at for the historical, the founding study, if you will. Okay. So what are we doing in differential scanning calorimetry? Well, this kind of looks like the guts of our ITC, doesn't it? So we've got a reference cell and we've got a sample cell, okay? But now we're not going to be injecting anything. We're going to put the sample in, and we got the reference cell. And now what we're going to be doing is starting to heat the two of them up, okay? And we're going to try to keep them always at the same temperature as we heat them up. But if we go through a phase change, there's going to be some heat in our sample cell, right? And so you're going to have to apply a differential heat to, or more or less heat, usually it's more heat because it's an endothermic process, to the sample cell to keep them always at the same temperature as you're slowly ramping it up, okay? So again, the beauty of these instruments is the detailed engineering that goes into carefully heating the samples, carefully monitoring the temperature, carefully adjusting that temperature. Okay? So the biological examples that one might study from this are protein unfolding, DNA melting, phase transitions of membranes. Okay? This is quantified by the temp TM. So here's what the data looks like. Okay? This is differential power, okay, translated into kilocalories per mole. Okay? And so we're going along, and then all of a sudden we get this rise. Okay? And at this point, we're kind of poised between our two phases. We have equal amounts. Okay? Equal amounts. Ratio gives 1, so K is 1, so delta G is 0 here at the maximum. Call this TM, the melting. And then we go beyond that, and now we shift over to the higher temperature phase, the unfolded phase, if you will. And then we come back down to a baseline here, which is different than our initial one. Because now we have a different system. We have an unfolded protein. We have melted DNA. And the difference here is our change in heat capacity between those two forms of the system. So here you get the heat capacity. You can get it directly out of the measurement. Okay. What are the parameters that we're interested in? This TM. Okay. This tells about the stability. Oh, you can get TM by other methods, right? You do melting curves with CD, melting curves with fluorescence. So you can get that. So that's a good way to confirm that this technique is matching other methods, okay? But you do get the enthalpy associated with this. Now that has to do with, all the bonds that are broken when you go through this phase change. Okay, the instrument, all right? It's the same box. It looks the same, okay? Here's our working differential calorimeter. George took this picture of his baby, all right? And notice it's in its own box that he designed and built to make sure that we can do these measurements under anaerobic conditions. It's a real working picture. We got the Kim wipes there. We got the tubes of things. But the most important thing in this picture is the little note at the top of the box. Sign the logbook, all right? Important lesson for all of us. Sign the logbook. Okay, know who used the instrument last? Did they clean it? What sample were they using? Okay, so very important. Okay, now here, remember, all we're going to do is put the sample in, and we're going to close it up. It's run at slightly higher pressure, so you can go to these higher temperatures without boiling the sample. Okay, but it is at constant pressure. Okay. And you can do that under anaerobic conditions. Okay, so what are we going to do here? All right. Well, actually, this is some of George's data. He's really kind of pioneering some of these studies. Insulin is a protein we've been studying quite extensively, as a lot of other people have. In my question to Fraser Armstrong about commercial applications of our field, turns out insulin is sold as a metalloprotein. Okay. When we synthesize it in our pancreas, it's packaged into little vesicles that are filled with calcium and with zinc. And insulin, in the presence of zinc, forms this hexamer. Here we are looking right down the threefold axis. Here's 3 insulin subunits. There's 1 zinc on top. There's 1 behind it. So zinc stabilizes it in this hexamer form. When we release insulin into our bloodstream, or then it dissociates into the monomer. This is the active form that binds to the receptor. Okay. So turns out if you take insulin under normal concentrations that we study, it's predominantly as a dimer, and you do the DSC measurement. Okay. Now, George has normalized the baseline, so you're not going to be able to pull out the change in heat capacity here, but you can see the TM and integrate the area and get the enthalpy of unfolding. So now we do the same experiment with the hexamer, okay? This is still kilocalories per mole of monomer, so it's same basis, but now we see Tm has shifted over, and we got more heat. So the zinc stabilizes this structure to thermal stability. It also stabilizes it to proteolysis. It stabilizes it to fibrillation. Because if you take a sample of insulin and let it sit around, you'll form fibrils. Interesting story there. Okay. Now, in the vesicles, we actually have excess zinc. So what does that contribute to the stability of the insulin? And here's the data in excess zinc, probably about another five or six equivalents of zinc. Notice the Tm has gone up even higher and more enthalpy. Okay, so basically, zincs are binding to this protein. We're galvanizing the protein, right? We're covering it with some extra zinc, and that leads to additional stability. So here, the metal is actually perturbing the properties of the protein in a good way. It's making it more stable. Okay, when you release the insulin from your pancreas, all that zinc goes in your bloodstream. Okay, we deal with it. All right, it's there playing an essential role, and here we are measuring its contribution to the stability of this particular protein. Okay, one other case, another old friend of this field, is carbonic anhydrase, zinc metalloenzyme involved in equilibrating CO2 carbonate in our erythrocytes. It's a zinc enzyme, has this very knotted type structure with beta sheets and alpha helices. The one zinc ion is coordinated to 3 histidines, 93, 95, and 118. The crystal structure of the apoprotein is known. It's a very stable protein. You can take the zinc out, put other metals in, which George has on his poster. Very nice study of how that contributes to the stability. But I wanted to steal some of his data here to show you what the metal contributes to the stability of this protein. So here's the DSC measurements of the apo form, okay? And here's this DSC measurement of the zinc form, okay? We get a noticeable shift in the Tm just by having the zinc bound there. Doesn't perturb the structure much at all, but it kind of ties it together like a safety pin, right? Not much change in the enthalpy, though. So that says that most of the enthalpy, in fact, is due to the protein. Very little is due to the breaking of the bonds, of the metal histidine bonds. So this is kind of a really good example. Notice you could do a titration here, right? You could pull out a binding constant, okay? And some people do that with this technique. All right, so one last slide here to kind of summarize DSC. Measure the perturbation of protein thermal unfolding by metal ions, quantify the thermodynamics, correlate with other methods like of thermal stability, and now try to correlate the thermodynamics of binding with the thermodynamics of stabilization. So, I did fill up the 90 minutes, Nikolai, okay? And we'll acknowledge the folks that did the work. I want to point out Ann Spuchis, who's at ECU, Nick Grossamy, who's at Winthrop, Jolene Schuster, who's actually a graduate of Penn State Erie, the Barron's College. She was the first person that told me about Penn State. She loves this place. Colette Quinn, I point out, she is the head scientist with TA Instruments. If you buy one of their instruments, you'll meet Colette. She's good. But I want to really point out Molly Carpenter, who's here at the meeting, and George Lisi, and I showed some of their work, funding from the NSF, and for a lot of the studies we've done. And I'd be happy to take any last questions here before we go for coffee. Yes?
That was awesome.
Oh, thank you.
Sorry, but what if you have metal binding and protein-protein interactions? Would you ever be able to tease out binding constant in that case?
Okay. So.
Say a protein, that's what I work in, is transferred between proteins. So what if you have a protein transferring metal to like a protein and they do interact?
Oh.
Would you be able to tease out anything other than?
So everything going on is going to contribute to your heat. So presumably the measurement you might be describing is you have one protein with a metal in the cell. you titrate in the other metal, okay? So you have to think about all the things that are going on, okay? So protein-protein interaction, as you mentioned, metal release from one, metal binding to the other, okay? The question is, could you measure some of those steps independently so that you could know how much they contribute to the total heat? So there you have to kind of put your hat on and think about, okay, so could I start with maybe the protein that has the metal in it and do maybe one of these chelation experiments, okay? And maybe do that with the other protein, and then at least know the metal binding and release, and then factor that in, and then what's left over is the protein-protein interaction. Would that work?
Yeah, so you could constrain it by having that information.
Yeah, bootstrap your way to the answer. Yeah? Okay, that's good. Yeah?
Can you use ITC if you're titrating gas?
Is a gas. Ooh, that's tricky. We thought about, we'll use ITC and we'll titrate in oxygenated solutions and we'll measure the thermodynamics of, cooperative oxygen binding to hemoglobin. But that's very tricky. There are other techniques, other calorimetric techniques, I'm blanking on it, that actually are better for gases. I think it's thermal acoustic measurements that you can measure the heats of gases binding. So if that's your interest, there's another method which I don't know about, but I can steer you to somebody who does. You probably have to collaborate with them because they may be the only person that does it, but that's gases are important in this field. Oh yeah, sorry, way back. Yes. Ah. Okay. So those pictures were taken with the front off. Okay, so we designed them so you can take the front off and so do all your work aerobically for, things where you don't need. But when you want to do anaerobic, you put the front cover on, and then we have these pretty small ports. It doesn't have to be very big because you're just taking samples in and out, and there's a little mini chamber, and it's a pretty small box, and that's for the advantage. You can purge it fairly quickly and get it set up before you then. And then, prepare the samples in the glove box, in seal containers, introduce them there, get it all aired, and then you got the gloves in just the right position so that you can load your sample in there. So, yeah, maybe as the field picks up, we will start selling them out of our garage, right? Yeah.
Are there any other questions? Well, I can say this is very helpful because there are many people who believe that you can just sort of inject your titrant into your protein, get a number out of nitric paper. There are all these controls that we haven't even thought about.
Yep.
Valuable information. Just out of curiosity, are there buffers that you would suggest people just completely stay away from when trying to do this sort of technique? Or,
Yeah.
Go ahead.
Yeah. So phosphate is one to avoid because metal phosphates tend to be fairly insoluble. There are a few special cases. Copper does a redox reaction with Hepis buffer, so you want to avoid that.
About tris, entomology in general, when we deal with metals, people always talk about
Be careful, tris. Tris has been good to us. I would say, and particularly with copper, it binds fairly tightly, so you can measure sites that have a fairly high affinity with that. But whatever the buffer is, you really need to know, its protonation enthalpy. You need to know its interaction with the metals, so that you can factor out that interaction that contributes to your signal. This post hoc analysis. So that's where we earn our pay.